Hello everyone, I Assistant Professor Pragati Shete from Department of Microbiology, KJ Somaya College. In this online lecture, we will study about the second type of the hemorrhagic fever causing virus called as the dengue virus. Alright, we know the dengue which is caused by this virus. Okay, so we will start our lecture with the structure and the family classification. So this virus will come under the family called as the Flaviviridae. Okay, the FVD is also used to denote Flaviviridae family with genera called as the Flavivirus. Virus name dengue virus. Okay, also known as the break-born fever. Break-born fever. Then it is most prevalence rate of this virus has been shown in the Asia, Caribbean countries, then the Pacific, West Africa and the India in the east coast. Okay, then morphology, if we come into the morphology, okay, then you can see in the structure of the dengue virus, then on that you can see the envelope. So it is the envelope virus. Okay, on the surface envelope it will have the envelope proteins. After the envelope it will have the transmembrane envelope proteins. You can see there are the red color rods, and above the red color dots are present, the circles. So that rods, that red color dots denote the transmembrane envelope protein. Transmembrane that will transfer the materials from the membrane to the outside. Okay, these particles will help the virus to penetrate into the site of the infection and with the help of the multiplication. Okay, then after that there is the membrane. Okay, there is the envelope, that is the membrane. Beneath the membrane there will be the matrix proteins called as the MA. After matrix there will be the capsid. What is a capsid? It is a protein coat, okay, which will protect the genome of the virus. So you can see there is the blue coloration lines in the triangular form. It has been shown, that is called as the capsid. Then the genome is the single stranded positively sense RNA strands. Okay, the virus, the size of the virus is 40 to 50 nanometer in the diameter. Okay, then the genome is bound by the nucleocapsid, right? As we mentioned, the around is the capsid, and inside the capsid there will be the single stranded positively sense RNA strands. Okay, and in the RNA there is the yellow color one, you can see a reverse transcriptase enzyme present, which will help for genome transcription. Okay, then along with the RT, the reverse transcriptase, the protease enzyme is also present in the virus, dengue virus. Okay, so this is the morphological characteristic properties of the virus. Now we will see the serotype. So there are the, and one more point, this morphology is very much similar to the yellow fever virus. Okay, now the serotypes in the dengue virus, they have 4 serotypes: den 1, den 2, den 3 and den 4. Now vector for this, now as we all know, the virus or the dengue we get, the infection we get due to this mosquito, right? Mosquito called as the Aedes aegypti. Okay, so this will cause classical dengue fever, dengue hemorrhagic fever and the dengue shock syndrome. So this virus will be responsible for causing all of the 3 diseases, 3 infections, 3 diseases. Okay, the classical dengue fever, then the dengue hemorrhagic fever and the dengue shock syndrome. Now first one, okay, first we will study about the classical dengue fever. Okay, that is the normal in general dengue fever that we get, the primary. Now this will mostly affect in the children and in the adults. the older children's, okay, in the teenagers and the adult, then bite from the infected mosquito will infect in the host cell. Now the infected blood of the mosquito will be having the virus within it, right? So that blood will be getting transferred into the host due to the bite of a mosquito. Therefore, the virus particle will be transferred via the vector into the host cells, host body. Bloodstream. Now the biphasic fever, that is called as the saddle back, headache, pain in the muscles and the bones. These symptoms are mostly observed in case of the classical dengue fever. Which are the symptoms? The biphasic fever, headache, muscle pain and the bone pain, okay, pain in the joints, right? Then the sleeplessness, restlessness, pain in the bones. So these symptoms are mostly observed. Then incubation period for classical dengue fever is the 5 to 8 days after the infection, after the entry, okay. The incubation period is 5 to 8 days. Now the maculopapular, okay, maculopapular rash will appear on the body on 3rd to 4th day, okay. Then febrile illness will remain about the 10 days. The febrile illness will be remaining latent, okay, and that is for about 10 days. Then complete recovery is possible in case of the classical dengue fever, and the disease is very rarely turned into the fatal one. Otherwise, this type of the fever is 90% or the 99% recoverable, completely recoverable, okay, and very rarely it will turn into the fatal disease, okay. Now we will see the second, that is the second type, that is the dengue hemorrhagic fever, DHF, and the dengue shock syndrome. So both the syndromes are very serious type of the diseases, and they are very harmful, they are very painful, okay. These are hyperimmune responses will be produced in this. Then these are mostly confined with the children, okay, mostly affected in the children of the 5 to 10 year old, okay. Now these cases will be present where there will be the multiple continuous cases of the classical dengue viruses, okay. If a particular area had the dengue patients, okay, previously, say 4 to 5 patients were there, and that get recoverable, but the dengue virus is still will remain there, right, because the vectors may present. And in that cases where there will be the multiple cases of this previously dengue fever, there that area will be more affected for getting this dengue shock syndrome or the dengue hemorrhagic fever, okay. This type of the syndromes or this diseases are very much harmful and the painful as compared to the classical dengue fever, okay. Then this is observed in the patients which are previously infected with the dengue virus, right. So this is also important point, that this syndromes are observed in the patients who are recovered or who got the previous dengue virus fever, okay. Now we will see the clinical features or the clinical signs, symptoms of this disease. Now, as we all know, in the dengue disease, the most common is the fever, right. Then fever, abdominal pain, the pain in the bones, pain in the joints, Okay, then retrobular pain, headache, conjunctival infection. Okay, then pain in the back and joints, right, as I mentioned. Then lymphadenopathy can be present. Okay, that is infection in the lymph nodes. Okay, lymphadenopathy. Then maculopapular rashes, the rashes will be present in some cases. Among this, the most common symptoms for the dengue virus disease are the headache, joints pain, and the fever. Okay, these are most common symptoms. Now, after the clinical features, the person will be proceeding for the laboratory diagnosis, right? Now, in the laboratory diagnosis, what is our first step? Our first step is to collect the sample. Okay, sample or the specimen. Now, the sample we should collect is the serum sample. Okay, or the blood sample. Now, serum sample that we will collect will used for this antibody and antigen detection. Okay, antibody and the antigen detection. And the sample that we collect, that is a serum, then plasma, or the whole blood. Okay, whole blood, in that also we have to remove the buffy coat. Okay, that we do with the help of centrifugation. Okay, then we can take the autopsy tissues, mosquitoes collected in the nature. Okay, so from there we can isolate this virus. Okay, we can isolate the virus, we grow them on the tissue cultures, on the cell cultures, and then we verify that, or we isolate, we quantify them using the PCR, right, polymerase chain reaction. These are the serological tests. Okay, now next one is the hematological diagnosis, that is the blood diagnosis. Now in that, thrombocytopenia, in the thrombocytopenia, where the blood cells will be 1 lakh or less than 1 lakh per millimeter cube, which is not optimum. Okay, and then hemo concentration, that is the concentration of the blood, all right, in the percentage. So that we have to determine, we have to check. After that, the microbiological diagnosis, and then the serology. Now, in the antibody detection, we can use ELISA. Okay, in case of the antibody, we can use the ELISA, and in that also the IgM antibodies and the IgG antibodies. Now, IgM will be producing very soon after the person will get the infection. Okay, then the IgM will be 5 days after the onset of the disease and persist for 1 to, that means it can remain for about 1 to 3 months, whereas the IgG will produced afterwards. Okay, and that will be produced in the higher concentration, and that will remain also for later stages. Okay, so if IgM is getting diagnosed, we can say early infection. IgG is getting diagnosed, we can say the latent infection is present. Then a strip of immunochromatographic test, that is called as the rapid test, that will fall under the rapid test category. And with the help of that strips, we can determine the IgM antibodies, since the strips for immunochromatographic strips are available for IgM antibodies. Okay, then in the antigen detection, we can determine an S1 antigen, antigen present on the structure of this virus. You can see in the previous slides, okay, there the nucleocapsid surface particles are present, okay, so that we can determine the NS1 antigen can determine. Now this can also be determined via the immunochromatographic test, okay. It is very rapid, it can complete in the 15 minutes only. Now this test should be done before the antibodies will appear, means the first day of fever, okay, this test must be done, because once the antibodies will develop, then this test will be not specific, okay. Then next, the isolation of virus. Now as we have discussed in the cell culture, in the tissue culture, so the isolation will be inoculate virus into the mosquitoes, cell lines will be produced, or the suckling mice, in that mice also the virus can be getting isolated. Then further identification by the fluorescent antibody test, okay. So in that, what is happening, the sample from that virus, from that mosquito or from that mice will be taken, and that will be used for fluorescent antibody detection. So if the antigen and the antibodies complex will be produced, then what will happen, the fluorescence will be emitted, which determine the positive reaction, which will confirm the isolation of virus. After that, we can use the PCR test, okay. After isolation, we proceed for PCR. Now the RNA can be detected, right, in the PCR. The genome can get multiplied, can get polymerized. So the viral RNA can be detected in the clinical specimen by RT-PCR method, reverse transcriptase method, okay. If you remember, reverse transcriptase is the enzyme that is present inside the genome of this virus, okay. Then viral genomic can be therefore detected, okay. Now the treatment, in this also the symptomatic treatments are available. We have studied in the Ebola virus disease. In the Ebola virus disease, there are symptomatic treatment available, right? So similarly, in this also we have symptomatic treatment for dengue virus. If the infection is severe, okay, then they infuse platelets to reduce the hemorrhagic manifestation, okay. Now in this also, there is not particular vaccine available against the dengue. Elimination of the mosquitoes is our foremost duty, okay, in order to avoid this type of infections, this type of diseases, right? Because the mosquitoes are the primary host, are the vectors, sorry, these are the vectors for these viruses, via which the virus is getting transferred, right? Therefore, we should control the spread of these mosquitoes. We should use the proper insecticides, the chemicals treatments, right, to reduce or to remove the population of these mosquitoes. Which mosquito? Aedes aegypti. Then to avoid the hemorrhagic fevers and the dengue shock syndrome in the immunized persons, a live attenuated vaccines containing all the serotypes of the dengue has been developed, and this vaccine is currently undergoing the trials, clinical trials, right? Then if the clinical trials will be successful, then the vaccine may be available, okay. This vaccine is for the dengue hemorrhagic fever, alright, or the dengue shock syndrome. This vaccine is for all 4 serotypes of the dengue, that den 1, den 2, den 3 and the den 3, den 4, right? The classical dengue fever is completely recoverable disease, okay, and that is not become fatal. In rare cases, that classical dengue fever will be fatal, otherwise it is completely recoverable, okay. This is the prophylaxis of the dengue virus disease. Now in this lecture we have saw structure of the dengue virus, then structure, then we saw the serotypes, the family, genera, then the clinical features, right, the clinical symptoms. Then we saw 3 types: classical dengue fever, dengue haemorrhagic fever and the dengue shock syndrome. Then we saw the morphology, we saw the structure of this dengue virus. After that we saw the laboratory diagnosis of this dengue virus, right? In the laboratory diagnosis, the antibody detection and the antigen detection, okay. After that the treatment, and then the prophylaxis or the preventions that we should take, okay. I hope you understood this online lecture. If you have any doubt regarding this, please ask in the comment section. Thank you for watching.